Hello friends. Today we are going to study about a topic called EEG signal, introduction and concept. Myself, I am Dr. Sarin Maitri. I would like to make you know what is meant by EEG signal and how an EEG signal is being used or how an EEG signal is being captured from the human brain with the help of a machine called EEG machine. Especially we usually see these EEG machines in the hospitals where it is used by the doctors or the lab technicians in order to capture the EEG signal. So now let us see how an EEG machine is being manufactured and what is the basic and origin of this particular EEG signal. Introduction. First of all, EEG signal is a neuroelectrical signal. Why? Because why do we call it as a neuroelectrical signal? Because all the neurons or the neural signals are the electrical signals. So it's a neuroelectrical signal firstly, and the neuron is a basic working unit of the brain. Neuron is a basic working unit of the brain. And what is this neuron? Neuron actually it is a specialized cell designed to transmit information to other cells or other glands or other muscles. So it is mainly used to transmit information from one part of the neuron to another part of the neuron or one part of the brain to another part of the brain. This brain is a kind of a structure or a kind of a part of a human body which has got a large number of neurons. Nearly this particular brain has got more than 10 billions of neurons which are present in a human brain. So now coming to the neuron structure has got basically three different kinds of parts. The first one is called as a cell body, which is also called as a soma, and the second part is called as an axon, and the third part is called as a dendroid. So now here you can see here now in this particular figure which has been given over here, we can see dendroid is given over here. This is one of the part of the neuron, and the axon is given. This is another part of the neuron. So now here, apart from this one, you can even see a small opening will be there at this end of the dendroid, and that opening or the gap we call it as a synapse. So now through this synapse only the information will be passed. So we'll try to understand that particular how the information is passed through the from now one neuron to another neuron in the next coming slides. And the other part is that I have even mentioned over here one amplifier has been given, electrodes have been given. So why do we need an amplifier? We need an amplifier. Basically, we need it should be like a low noise amplifier. Why do we need a low noise amplifier? Why? Because firstly, the neuron is a kind of signal. Neuron signals is a kind of signals which are of low amplitude and low frequency band. So that's why in order to amplify this particular small frequency signals or small amplitude signal, we need an amplifier we should almost eliminate the noise which is present, which can be due to the noise due to the blood flow in the body or due to another activities which are surrounding around that particular area where we are trying to capture this particular signal. So now here we need a low noise amplifier which is mainly used to magnify these small signals which are of low amplitude and low frequency from the using an extracellular electrode. So these electrodes are mainly used to capture that particular signals from the cortex part or the scalp of a human brain. So now this is how this particular amplifier will be working. Now coming to the next slide, in the next slide we'll try to see the origin of EEG. How does EEG signal works and how that signal has been captured. The dendroid you might have seen here in the previous slide, in this particular slide, dendroid, it looks like an antenna. So here that's why I have defined this dendroid as a dendroid is an antenna like structure part through which a neuron receives the information from one cell to another cell. And coming to the second part of the neuron, that's an axon. If you see the figure over here, axon is a long structure here. So that's why the axon is a long cable-like structure which can be of even 1 meter long length. So that's why the definition of this axon can be axon is a cable-like structure of neuron through which information is sent from one neuron to another neuron, and the length can be almost like 1 meter long. Then coming to the nerve cell in the brain produces these signals that are called as an action potential. Whatever the nerve cells produces the signals in the brain, which means that the brain cells or the brain nerve cells will produce a signal, and these signals we call as an action potentials. These action potentials move from one cell to another cell. These are the signals which we are supposed to capture from the uh, machine called as EEG machine. So now these action potentials move from one cell to another cell through a gap called as synapse. So now here you can see here in this particular dendrite, this dendrite will be having an opening at this end of the part. This opening this opening we also can call it as a gap, and that gap is called as a synapse. Through this gap, the information that is called as an action potential signal is been passed from one signal to another, from one cell to another cell. And apart from this one, we also find chemicals which mainly help in order to move these signals from one cell to another cell. These chemicals we call as neurotransmitters. Neurotransmitters are the chemical, special kind of chemicals which help the signals to move from one neuron to another neuron through this gap called as synapse. Basically, there are three different kinds of neurotransmitters. The first one we call it as an excitatory neurotransmitter. What is this excitatory neurotransmitter does? The excitatory neurotransmitter is the one which will help the action potential or the signals through move from one cell to another cell. And coming to the next one. Okay, before going to the next one, let us see an example of this excitatory neurotransmitter. The example of excitatory neurotransmitter is a glutamate. And coming to the next kind of neurotransmitter, it is called as an inhibitory neurotransmitter. Inhibitory neurotransmitter is a transmitter which will try to stop the motion or movement of the information or the action potential from one nerve cell to another nerve cell. So this is one kind of the neurotransmitter chemical. That the example case is a GABA. Then the third one is called as a neuromodulator. You can see that the name itself, it tells that it does some kind of modulation. It is a neuromodulator which is mainly used to modify the information which is received from the past synaptic cell and sends it to the another neurotransmitter. So this is how this particular information is being passed from one neuron to another neuron through a chemical called neurotransmitters. Now coming to the EEG. EEG is called as in the full form of the EEG is an electroencephalogram, which is a recording, which is nothing else but it's a kind of machine, or it's a signal which is captured by the machine, or else we can call it as a recording of an electrical activity of a brain from the scalp. EEG records the potential difference between the two electrodes. So see now. In this particular diagram, you can see that on the scalp of this particular person, there are two electrodes, and the two electrodes will not be getting the similar kind of EEG signals. It can be of different kinds of EEG signals with difference in the amplitudes or difference in the voltages. So now these two electrodes will be capturing two different kinds of potentials, and those two different potentials have been captured by these electrodes and have been given as an inputs to the amplifier. And once this amplifier gets this inputs from the electrodes, this amplifier will be doing a difference in these two amplifier potentials, and will take the difference of these two potentials as an input, and it will amplify that one, and it will give the output of that particular signal. So this is how EEG machine will be working. EEG machine will be having an amplifier. That amplifier will be doing, or it does the difference of the two potentials, and it takes the difference of two potentials and gives us an output waveform. This activity is influenced by an electrical activity from the brain structures underneath the cortex. And you can see here one example where you can see this is a one kind of EEG machine which is kept on the scalp, where from this electrodes the signals have been captured. So this is how the EEG machine will be working. EEG machine. Just now I spoke that EEG machine will be using a difference amplifier. This difference amplifier will produce each channel or each trace of the activity. Each amplifier will be having two inputs. These inputs are nothing else but which are connected to the two different electrodes, which we have seen in the previous slide. The two electrode information will be given as two different inputs to the amplifier, and that amplifier will be amplified, taking the difference of the two inputs, and will be amplifying that particular small signals, and it will be giving us an output as a EEG channel. An electrode is connected to the each of the input. The difference amplifier will measure the voltage difference or the potential difference between these two inputs, between the two signals at the each of its inputs. Then the resulting signal is amplified and it is displayed as an EEG activity. So this is how an EEG machine will be working. It means that it's totally electronic device. EEG machine is an electronic device which is mainly used to capture the signal from the human scalp of the human brain or the cortex part, and it will does the difference of the voltage signals, and it will give as an one signal which is amplified one. That signal we call as an waveform of the EEG or EEG activity. And coming to this particular table, you can see here now there are different kinds of signals have been mentioned. These are nothing else but the different EEG bands or different EEG signals. There are basically four different types of EEG signals which can be captured from the scalp. First one is called as a delta, second one is theta, third one is alpha, and fourth one is beta. Delta, the amplitude of this delta is 2 to 100 microvolt, and it falls in the frequency of 0.5 to 4 hertz, and this particular signal can be captured whenever a person is in the deep sleep. Coming to the theta, the voltage amplitude is 80 microvolt, and the frequency is like it falls in the frequency band of 4 hertz to 8 hertz, and this signal can be captured whenever a person is in deeply relaxed condition. And the next one is alpha. Alpha will have a amplitude of 50 microvolt, and it falls in the band of 8 hertz to 13 hertz. And this particular alpha signal or EEG signal can be captured when a person is in awake or relaxed condition or closed eye condition. And the fourth one is a beta, that's the final one. Beta has got an amplitude of 20 microvolt, and which falls in the frequency band of 13 hertz to 30 hertz, and this signal can be captured in this particular conditions of a human body, that's an anxiety dominant condition or active mind condition or busy condition. So these are different kinds of EEG band signals. So this is how an EEG signal will be working, or this is how an EEG signal can be captured from the scalp of a human brain with the help of is the electronic circuit which is called as a differential amplifier. So based upon this one, the research is going on in order to come up with the more efficient sophisticated neuron amplifiers, which we can find in the present day research. Thank you, everyone. I hope the particular topic is very clear to you, and I hope I have given some insight onto the biomedical part of the electronics, that's the bioelectronics. Thank you, everyone. If you like this particular lecture, please like the video, subscribe the channel, and also share it among your friends so that we can come up with more interesting topics on this electronics. Thank you, everyone.